Hello everyone, my name is Alia, and in this video, I will be explaining and conducting the alcoholic fermentation in yeast experiment. Glucose is a type of carbohydrate. It can be catalyzed into pyruvate through glycolysis. Now, these pyruvate molecules can undergo anaerobic or aerobic fermentation, but we're only going to focus on anaerobic fermentation for now. In this type of fermentation, anaerobic organisms such as yeast will break down the pyruvate into ethanol and carbon dioxide without the presence of oxygen. The objective of this experiment is to observe fermentation activity of yeast and find the rate of fermentation of yeast in different types of carbohydrates. The materials you're going to need are 4 plastic bottles, 4 stickers for labeling, a bunch of balloons, baker's yeast, a teaspoon measure, and the carbohydrate sample I'm using is table sugar, glucose syrup, and cornstarch. Lastly is warm water. The water needs to be warm or at 37 degrees Celsius to be precise. I use a thermometer. First and foremost, I put on my lab coat neatly. Then, I label all the empty bottles from number 1 to 4. I also included the initial letter of the sample I'm using into the label so I don't confuse it. Then I filled all the bottles with 10 milliliters of warm water. Then I added 3 teaspoons of the carbohydrate sample into the bottles accordingly. Bottle 1 is table sugar, bottle 2 is cornstarch, bottle 3 is for glucose syrup. This process was quite messy because the spoon was too big and the opening on the bottle was too small. Glucose syrup is the hardest to work with because it's very thick and viscous, but I managed. I did not add any carbohydrate sample into bottle 4 because it acts as a controlled sample. After that's all done, I added 1 teaspoon of yeast into each bottle very carefully not to make a mess. Then I mixed all the contents by closing the opening using my thumb and shaking the bottle gently. After it looks homogeneous, I covered the opening of each bottle using balloons of the same size. Finally, I put it in a warm spot and observed what has occurred in the bottles and the change in the size of each bottle every 5 minutes for 25 minutes. During this process, the yeast will undergo anaerobic fermentation by breaking down the sugar molecules into carbon dioxide and alcohol. The production of carbon dioxide gas will slowly inflate the balloon in the experiment. Therefore, yeast activity can be measured mainly by observing the size of the balloon and the bubbles that can be seen inside the bottles. In brief, fermentation by glucose syrup should inflate the most because it is a monosaccharide, but it seems that table sugar supported the fermentation more readily than glucose syrup. And then it is followed by cornstarch, which has a very slow fermentation rate, and then culture sample has little to no fermentation. A very similar process happens as bread rises. Carbon dioxide from yeast create thousands of air bubbles in the dough. This is what gives baked bread its airy and light texture. I hope you guys understand and enjoyed this video. Thank you.